Our first presenter is Anish Pyne from Mellon College of Science, whose 3-minute thesis is entitled "Practice Makes Pathogens: How Bacteria Train for Infections."
Dangerous bacteria don't just appear in hospitals. They're constantly training for infections in places as ordinary as the water pipes in this building right now. One such bacteria, called Pseudomonas aeruginosa, or PA for short, causes chronic and life-threatening infections in people with cystic fibrosis, multiple wounds, and in hospitalized patients. Now, these infections are extremely difficult to treat because this bacteria, in most cases, exists as biofilms in these infections. Now, what are biofilms, you ask? So biofilms are these large aggregative communities that act as fortresses, shielding these bacteria from external threats like antibiotics or our immune system. But here's the mystery. Even before this bacteria enters our body to cause an infection, they actually live in our drinking water system. Now, this environment is a harsh one. There's almost little to no food, and the bacteria has to fight constantly in order to survive. This led me to ask a very simple, yet a very important question. Does surviving in this drinking water system prepare this bacteria for an infection even before they enter us? And to answer this question, I recreated the drinking water system in my lab and allowed this bacteria to evolve over time. As they continued to adapt, we observed several genetic changes that promoted the survival of this bacteria in our drinking water model. But surprisingly, we observed this one gene that kept on getting mutated again and again and again, which led us to believe that that gene must have a specific role to play in regulating the survival of this bacteria in this model. And when we removed that gene, we saw that indeed the bacteria grew significantly weaker. They struggled to form biofilms, and they couldn't even defend themselves against oxidative stress, which, by the way, is a key weapon used by our immune system against these bacteria. So in summary, my thesis so far has unearthed a gene of unknown function in this bacteria that we now know regulates biofilm formation and survival of this bacteria in the drinking water environment. So this discovery can change the way we think about infections currently, because understanding where and how the bacteria trains for infection might actually be the key for us to develop newer strategies to prevent an infection even before it occurs. Because when it comes to the fight against infectious diseases, understanding where and how the bacteria train can actually be the key to prevent where they strike. Thank you.